this special episode of Voice of the Sea, we're traveling from space to the deep sea with Exports, a NASA and National Science Foundation project that brings together researchers from around the world to combine new technologies in at-sea research, satellites, and mathematical modeling in ways that will greatly advance our understanding of current weather patterns and longer-term climate change. A critical part of Exports involves understanding the relationship between phytoplankton at the ocean's surface and carbon transport to the deep sea. As we shared in previous episodes of Voice of the Sea, the Exports team recently completed a series of studies in the North Pacific Ocean Basin. Exports team members are now preparing to head to sea in the North Atlantic, where they will study seasonal phytoplankton blooms. Meanwhile, NASA's new PACE mission to record ocean color in high resolution from space will greatly improve scientists' ability to make remote observations. Engineers are in the final stages of designing and building the PACE satellite, whose images of ocean color will help scientists identify species of phytoplankton and find areas of high concentration. We talk with NASA scientist and PACE project member Jeremy Werdell, who works at the NASA Goddard Space Flight Center in Maryland. But first, we start off talking with engineer and PACE's mission leader, Gary Davis.
My background is engineering. It's propulsion engineering. If you think of the fuel tank and the small thrusters that are on the spacecraft, that's what I started on at Goddard. And then from there, kind of graduated to take on the whole spacecraft. And now I've got the whole mission.
When are you planning to launch?
We think we're probably going to launch the end of 2023, maybe early 2024. We are launching from a SpaceX Falcon 9, and we will be launching from Kennedy Space Center. The Falcon will do a direct ascent for us. So the fairing will come off roughly 2 minutes after launch, and then we get separated about 21 minutes after launch. So it's not very long before we are flying free by ourselves. And so our first critical activity that we'll watch is to make sure that the solar array unfurls, because it's been folded up. And then the spacecraft will want to figure out where the sun is and point the solar array to the sun so we can start charging up the battery. Imagine if you're in space and if you take out your left arm and put it up, that's your solar panel. So the sun is always on that arm. So that's nice and warm and you're getting all your power. The right side of your body is cold because it's always in the shade. And we want to do that because the detectors of the instrument have to be cooled, very cold, for them to be efficient. So we have radiators there that always see deep space, so they're keeping us cold. And then in front of you, you can imagine you have a very strong shield, because we want to shield all of our spacecraft from micrometeoroids or space debris. So we have a big shield there. And then in the back, protected by the shield, is the instrument that looks down. So if you can imagine looking down as you're running forward at 17,000 miles an hour over the ocean and twisting your head from left to right to scan across the ocean, that's what you see.
And then how long do you expect the spacecraft to live for?
Our mission requirements are to last for 3 years. We should last longer than 3 years, and we put enough propellant on board to last at least 10 years. We use the propellant for three things: to keep it in orbit. The second thing is, in case there's another spacecraft that might be in our path, we might need to maneuver around something someday, so we have it for that. And then at the end of life, after the mission has done all of its science, we have to have enough propellant that we can target a safe part of the ocean to deorbit the spacecraft.
So you're actually planning to recover the PACE satellite?
We won't recover it. We just target a part of the ocean where no one is, and we make sure it lands in the ocean. Most of the spacecraft is made out of materials that will melt upon reentry, so really there's not too much that would ever make it back to land. The observatory is roughly 1,500 kilograms. That's about as big as, say, an SUV or a passenger car. And we use kind of three different names for it. You can call it a satellite, or you can call it a spacecraft, or you can call it an observatory. We make sure that we can design a spacecraft that can fit all the science instruments that we want and give them enough power and data rate and all that, and hold them nice and still so they can take pictures of what they're looking at. And that whole thing together is called the observatory. The cost of the mission is probably, if I had to guess, I would guess around $980 million for the entire mission. That covers probably a 10-year time span from when the mission was first conceived all the way through getting the science data.
Can you give me an idea of the number of people who work in the teams that are preparing PACE?
Off the top of my head, I would guess around 250 people are probably full-time working on the project. If you counted probably every person who ever worked a few hours on this mission, it would be probably over 5,000 people.
And then once it is in space and it's able to send data, how many people will it take to manage it and keep it running?
Once everything is in stable operations, we have a control room that only needs maybe 2 or 3 full-time people. When we send up commands, we have enough commands that we can tell it to do things for about a 2-week period, and it'll be fine by itself. But that's just to control the spacecraft. On the other side, you have the scientists who are getting the data. And to be honest, I don't know how many folks they will have analyzing the data every day and looking at it. You'll have to ask Jeremy that question. All the data is completely public, and all the descriptions of how to interpret the data, that's all public. You can go online once it's operational and get all your PACE data and make your discovery.
May sound a little trite, but it's kind of evolved into, holy moly, we might actually pull this off. And if we do, the contribution outweighs any little thing I'm going to do scientifically. Everything we read about in climate change, this is going to be a game changer in understanding it. Far better capabilities of knowing where our carbon is going, how the ocean and atmosphere work together or work against each other, all of these things. The goal is to have 1 to 2 day global coverage, meaning we see every spot on the Earth every 1 or 2 days. These are polar orbiting satellites. It crosses the South Pole, ascends to the North Pole, goes over the dark side of the Earth, and then just keeps repeating. We designed the instrument to have a wide enough swath so that these little paintbrush strokes as it ascends north will have a little bit of a gap around the equator, but generally try to paint the next row over, so to speak. And it does this 16 times a day, I believe, which is enough to paint the entire globe. And then the next day it's phased, so those little gaps, it just fills in the blanks. And it goes without saying, too, even if you're not in view of land, you should care about phytoplankton, because I'm going to go out on a limb here and assume all of our viewership here really likes breathing and really likes eating. In which case, say thank you to your phytoplankton, because whether you eat fish or not, or seafood or not, they are all contributing to the base of the food chain and our oxygen.
The University of Hawaii's Sea Grant College program, focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
Next, we're talking with exports researcher and University of California, Santa Barbara student, Sasha Kramer, about her research on the relationship between ocean colors as seen from space and the type of phytoplankton in the water.
The work that I'm doing right now in graduate school is taking a combination of satellite measurements that are looking at the surface ocean on these really broad spatial scales, and then more concentrated samples that we take out when we go out on boats or at observatories or docks or things like that and trying to figure out which phytoplankton are in the water. Different groups of phytoplankton contribute differently to this carbon cycle or to the food webs. Some are more desirable to certain organisms to eat than others. Some are toxic to either animals or humans. So knowing which phytoplankton are there is really important, and being able to do that from space would give us a lot more information about the surface ocean on these really broad spatial and time scales. If you look at a satellite image and you can see a couple of different colors on the image, you know that that might be related to different types of phytoplankton. So then getting to have your ship out there at the same time as you're collecting that type of image, you might be able to say, oh, cool, the brownish reddish color we see here was this type of phytoplankton, and the greenish bluish color over here was a different kind. On the exports cruises, I've been part of the hydro team. They're taking hydrographic measurements to support kind of the whole exports project. Phytoplankton pigments and organic carbon, we're taking samples for both of those parameters. We also are looking at inorganic nutrient concentrations, which phytoplankton need to grow their cells, and some biogenic silica, which is something that's found in diatom cells. And we also are looking at a bunch of different methods of seeing dissolved organic carbon. So either through kind of seeing what color the water is there dissolved carbon that's changing the color, or just direct measurements of that. We know that phytoplankton do a large amount of the photosynthesis on Earth, but we also know that some groups of phytoplankton can do kind of a better job than others. Some of the really big cells that have kind of these heavier minerals that make up their cell, diatoms. So those are a big one that you hear talked about a lot. And those ones can take up a lot more carbon dioxide into their cell. And more typically, those are cells that will sink all the way to the bottom of the ocean and kind of make it into that sedimentary rock cycle. If there are more diatoms in the middle of the ocean, there's likely going to be more efficient carbon exports. There will be more carbon dioxide from the atmosphere going into cells, getting to the sediments. Alternately, if they're really tiny phytoplankton, so like cyanobacteria or green algae, those are just not as efficient at taking up carbon compared to their cell size. And when they do take up carbon, they're often eaten pretty quickly by things, or their cells just don't have enough weight to kind of sink through the water and stay in the upper ocean, and are either recycled by bacteria, and so all of that carbon goes back into the ocean and some of it eventually gets back into the atmosphere, or they're just eaten by other small things and then that carbon is respired out again. Knowing the balance of who is there and what they're doing is really important to tell us about if the ocean will still be this effective mechanism for taking carbon from the atmosphere and getting it to the deep ocean.
Next, Colleen Durkin at the Moss Landing Marine Laboratories in California explains how, on the Exports project, she samples tiny particles in the ocean using traps equipped with a special clear gel that helps separate and identify individual particles to make links between measurements made at sea with observations from space. So tell me about the types of samples that you were collecting on the research cruise.
I am part of the trap team, and we collect the sinking particles directly. What we do is we put out these instruments, these sediment traps that collect sinking particles, and then we recover it so we have a physical sample of what those particles are. Some of my collaborators are measuring the chemical contents of those particles, and what I specifically am doing is analyzing exactly what those particles are using microscopy. The way that my samples are collected, it's in this gel layer. So there's a jar at the bottom of one of the collection tubes that has this gel layer. And so then when the particles fall into the gel, they stay separated from each other and intact. And then the gel is clear, so I can just put it on a microscope and take hundreds of images of it and then identify exactly what is sinking. So what's the biological source of the carbon that's sinking. And then the second thing I do is I can physically extract an individual particle. So an individual marine snowflake, pull it out and isolate it, extract the DNA, and then sequence what phytoplankton are present inside of those sinking particles.
So what kind of tools do you use to take images of the particles?
There's a camera attached to the microscope.
Once you have the image, how do you go about identifying the particle or the plankton from that image?
That is a good question. It took me many years to figure out how to categorize these particles. They are all different kinds of fecal pellets produced by different types of zooplankton. They are ambiguous, fluffy forms of detritus. You can really hone in on these beautiful biological details, and those tiny molecular processes and microscopic cell biology actually has a huge impact on the whole world. That's why I'm doing oceanography, and I love that we get to mix all of these disciplines together.
We are looking for a few heroes, mentors, trailblazers, innovators, a passion to change lives, spark curiosity, open hearts, and awaken minds. Help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.
Next, exports researcher James Fox from Oregon State University takes us into the weeds of phytoplankton blooms to help understand their effect on the carbon cycle, which drives global climate patterns.
In late spring or summer, sometimes you'll have a big storm come through. Normally the water column is very stratified, so you have this thermally stratified layer at the top and very few nutrients. But occasionally you get a summer storm that will come through and it will mix up the water column, and nutrients will be available kind of throughout the whole water column, and the light field will change. And so people know that when you get that mixing event, lots of carbon or lots of phytoplankton get mixed down into the deep ocean, where they kind of get locked away for a long time. But people didn't understand that, they didn't really know what happened once that water column re-stratified, because phytoplankton get trapped down in the deep part of the ocean as well. And there might be enough light for them to survive and grow there, but we can't really measure that from space, and we don't really think about that too often. After the phytoplankton get mixed down to that darker area of the water column, they can still survive and they can still grow. It's only really with the recent addition of these autonomous platforms like floats, like gliders, it's giving us a really different picture of how phytoplankton behave over an annual cycle. So using all of these new technologies, maybe we can start to develop new models, or at least we can better understand what these measurements look like. That will help to inform our satellite models going forward.
Can you just give me a description of what happens in a phytoplankton bloom?
Yeah, well, that's a little bit of a testy question at the moment.
I thought it was going to be more straightforward.
As phytoplankton are coming out of the winter, the water column is mixing a lot, and that means that they don't have a huge amount of light. And also, because it's winter, the day length is much shorter. Although the phytoplankton in the upper water column have lots and lots of nutrients which they need to grow, they don't really have enough light. But as spring comes and you get less intense wind-driven mixing and you have more thermal stratification, the layer that the phytoplankton occupy in the top level of the water column starts to shallow, starts to move up into the water column, and it starts to get thinner. That means that the phytoplankton have much more light available to them, and so they start to grow quicker because they have lots of light and lots of nutrients. As they start to grow, they start to outpace their predators, and they also start to accumulate lots and lots of biomass, which is basically a phytoplankton bloom, and that's the thing that you can see from space.
Next, we're talking with Massachusetts Institute of Technology graduate student Shawnee Traylor about the sensors she is using in the Exports project to help improve models of carbon flow in the ocean. Shawnee uses autonomous floats and gliders equipped with chemical sensors that collect data in the ocean and report back to her by satellite.
My research is really interested in filling some of these information gaps that we have with respect to the global carbon cycle. really understanding all of the factors and all the variability in ocean carbon sequestration is going to be really important in projecting how that's going to change in the future. I'm a second-year PhD student in a joint program between MIT and the Woods Hole Oceanographic Institution, or WHOI as we call it. My background is chemistry, actually, but my work now lies more at the interface of chemistry and engineering. So a lot of the work I do, outside of exports is in the realm of chemical sensor development and how we can use these sensors to monitor the ocean health and studying these kind of natural biogeochemical cycles. A lot of what I focus on is how to best measure dissolved gases, such as oxygen, carbon dioxide, or methane, which can tell us a lot about the way that these elements cycle through a system.
What you see here are called biogeochemical Argo floats, commonly known as BGC Argo. We have temperature, conductivity, and depth here. So the temperature sensor is just a simple thermistor, so it's this long metal rod here that's a little bit hard to see. The conductivity happens in a cell, this long cell here. Up here is how it communicates to satellite via iridium. So every time it takes a profile, it comes to the surface and it phones home and sends us those data. Here we have our biooptical sensor, which allows us to measure things like chlorophyll, fluorescent dissolved organic matter, and the backscatter of light on particles. This is our oxygen sensor, which is an optical-based measurement. There's a solid-state pH sensor in here. And finally, here is our nitrate sensor, which measures nitrate via this optical window in here. These are some of the first floats going out. They're the first floats from our lab ever. Ideally, they'll be out for at least, on the order of 6 months or so.
Oh, wow. So a longer time frame than the science cruise itself.
Absolutely. So the benefit of all of these autonomous assets is that it serves to contextualize the broader cruise period by capturing these processes that happen, before and after. I tend to be the one doing all the calibrations in lab, so pre-deployment checks, often the post-deployment checks if we're able to retrieve and recover. And then I go to the lab with these water samples and I do what's called a Winkler titration to really precisely measure down to the fraction of a micromole how much oxygen is in that water. To be honest, I had never heard of oceanography. I was going to be a hairdresser. I actually dropped out of cosmetology school to go to college, but I was out there in the field deploying things over corals and diving, and when things broke, I was the one that had to fix them, and I eventually was like, hey, what's this called? Like, how do I keep doing this? And the professor that I worked with was like, well, there's a place called WHOI, where he used to work, actually. He told me about chemical oceanography, and I was like, great, that's how I can be a plumber and an electrician and an engineer and a chemist and, a pseudobiologist at times, and just kind of try to understand, all of these really fascinating disciplines.
And your hair looks great. Through its revolutionary technologies, collaborations, and cross-analysis, exports will generate new understandings of phytoplankton and the carbon cycle in the ocean, findings which are critical to predicting future climate conditions. All exports and PACE data is free and publicly available. Learn more at voiceofthesea.org. Follow us on social media at Voice of the Sea TV. Mahalo for watching Voice of the Sea. Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org.